Good day, everyone. My name is Janina Benso, and I work with the International Office of the Leprosy Mission. Thank you for joining us for today's webinar, which is being recorded, and we will make it available to you as soon as we can. I have the great privilege to introduce to you Professor Dr Annemieke Geluk, who is an immunologist from the Leiden University Medical Centre, Department of Infectious Diseases. Professor Geluk is a long-time friend of the Leprosy Mission and works closely with our research teams in Bangladesh and Nepal on a number of studies related to immunology and immunodiagnostics. Professor Geluk is one of the world's leading experts on the topic of leprosy diagnostic, and we are really pleased to have her with us today to tell us the latest about diagnostics. Professor Geluk will speak to us for about 30 minutes, and then we will have the opportunity to ask questions. Please put your questions in the question and answer section at the bottom of your screen. Thank you, Annemieke, and over to you.
Thank you very much, Janine, and welcome everybody. And as Janine already mentioned, I'm a chemist and immunologist, and I started working at the Leiden University Medical Centre on leprosy diagnostics in 2004. And before that time, I worked on more basic immunology of mycobacterial disease, leprosy and TB. So in the slides that you see in front of you now, depicted are the people from my group that are working on leprosy diagnostics for the past 4 years. And the research of my group has two important pillars, namely lab-based basic research, so that's immunological research, and on the other hand, there is the translation of what we find in the basic research to the field. So field translation, that's another big pillar, important pillar of the leprosy research. And the three persons that you see here are part of the core group that work on leprosy diagnostics in my group. And because of the vision that we have on scientific research for leprosy, that not only scientific, but also societal and philanthropic valorization to results from our research, we aim at research that is designed to predict, diagnose, prevent, and better treat leprosy by translation of that basic research of immunology to field applications. And therefore, what is required in our research is the efficient use of talent and resources, using a team approach, and using also or requiring global commitments. And therefore, currently, we're working together with several research sites and field sites on our development of diagnostic tests. And so we're working together in this global approach with, for instance, people in Brazil, in India, in Bangladesh, and in Africa, African countries. Well, during this webinar, I will discuss leprosy diagnostics and we'll address several topics. So first, why leprosy research is necessary and the challenges we have in leprosy and also the challenges we have in our research. Furthermore, development of the diagnostic tests that we currently have. Also, I will address the use cases for such diagnostic tests. And finally, future research. But first, I would like you to keep in mind that there are several things that leprosy is definitely not. So it's not a genetic disease. It's not very contagious. It is not deadly. It's also not untreatable, and certainly it's not eliminated yet. But what is leprosy? It is a poverty-associated, neglected tropical disease, which is caused by a bacterium called Mycobacterium leprae, and it affects preferentially the skin and the nerves, causing irreversible lifelong handicaps that are also frequently associated with stigma. Most cases of leprosy nowadays occur in India, Brazil, Indonesia, but also in Bangladesh and several African countries such as Ethiopia and the Congo, leprosy is still apparent in several niches. We can efficiently treat leprosy using multi-drug therapy in a combination of antibiotics, but to treat it efficiently, it has to be detected early. And this is not always possible because we're currently still facing several challenges in leprosy research. For instance, very prominently is that there is stable new case detection rate. So that means that over more than a decade, the same number of cases, about 200,000, is reported every year. And this number also includes 10% children. And it means that every hour, 22 new cases are being diagnosed and reported. And so this all indicates that the transmission of Mycobacterium leprae, the positive agent, is still ongoing and it's active. But the diagnosis that is mostly done is clinical. So it means the diagnosis is done at a later stage than the early diagnosis that we would want, that is essential to prevent irreversible handicaps. However, there are currently no tools and no tests for such early diagnosis or even to predict leprosy. So there is a gap, a knowledge gap, or as you say, a diagnostic tool gap between what we want and what is required to prevent the irreversible handicaps and what is currently available. And on top of that, there is another aspect of leprosy, which is very specific to this disease, and which is the long incubation time. And that also complicates the diagnostics. If we compare, for instance, the incubation time of leprosy to the incubation time of COVID-19, with several of you are, of course, now very familiar, that COVID-19, after infection, about 7 to 10 days after infection, people will get symptoms if they are going to get the disease. Whereas for leprosy, this time span is typically 2 to 6 years, but can even be longer. And as an example of such a long incubation time, depicted in this slide is the chimpanzee that was captured, was 1 or 2 years old, from Sierra Leone in Africa and brought to a zoo in Japan, where it showed skin lesions at the age of 30. So this indicated, because there's no active leprae transmission in Japan, this indicated that the animal already contracted the bacteria 30 years before it developed clinical symptoms. So this complicates diagnostics, and this is something that we as leprosy researchers need to keep in mind. And what happens during those, 2 to 6 years, which it takes typically for leprae to provide clinical signatures in a human being? Well, after transmission, usually in 99% of the cases, Mycobacterium leprae is killed, and nothing happens to the person that has been infected. But in some cases, Mycobacterium leprae can survive as a sort of hidden in the body, and it would take 2 to 6 years before clinical symptoms occur. But in that time, Mycobacterium leprae can be transmitted, or a person can start developing a disease. So it is very crucial that for that stage in which Mycobacterium leprae is living in the person and can be transmitted, that stage is detectable through diagnostic tests. So we can treat early, either treat with post-exposure prophylaxis if the person doesn't have disease yet, but to prevent further spread, or treat early stages of the disease. So having diagnostic tests for that stage is very important. Well, we work at the LUMC on immunology, and we translate that basic immunology to diagnostic tests. So why do we use immunodiagnostic tests, and why not use like a PCR, for instance, which is also, as you're familiar with through COVID, which is also used to detect the pathogen causing the disease? Well, after a pathogen infects a human host and invades the host, a lot of mechanisms start. So the immune response is activated, and thereby all kinds of cells are activated that are trying to kill and eliminate the pathogen from the body. And these cells that are activated, they produce molecules like cytokines, chemokines, or antibodies. And these are called biomarkers. So these molecules that are a kind of footprint of an infection or of disease that even remain visible in the body when the pathogen has been cleared. So when the person has, for instance, become PCR negative. So we use those biomarkers as this footprint for infection, and immunology, so the immunological biomarkers, allow us to do so even in the absence of the pathogen. What's important then to keep in mind is that all phases of infection may have different biomarkers. For instance, the first phase would be that a person that Mycobacterium leprae is transmitted to a person, but a person is able to eliminate the bacteria without infection being started. And this is usually done by innate immunity. So the biomarkers are derived from innate immunity markers. The second stage, which could occur, is that a person is really infected, but that the infection is then eliminated through the activation of T cells, so-called adaptive immunity. And these have different biomarkers associated with that stage. And a third stage would be a stage in which the bacteria are replicating and a person is not able to contain the bacterial growth, and that there is still no really clinical disease. So there is replication of bacteria, but it is at an asymptomatic level. So we call that actually early disease. And this, again, has different biomarkers associated with that stage. And finally, there is a lot of replication of bacteria, and there is clinical visible damage of the host. So that is real full-blown disease. Of course, there is leprosy, also the occurrence of co-infection in leprosy patients, for instance, Helminth infection, but also diabetes, and also environmental factors like food or the environment, tropical areas or dry areas. That could all play a role in the biomarkers that can be detected in an individual. And furthermore, leprosy also is a very complicated disease because it does not have one type of disease. As you see in this slide, leprosy has a disease spectrum. And on one side, there are individuals that can contain the bacterial growth and are having only small numbers of bacteria. And this on the left side, the falcibacillary cases. Whereas on the other side, in the multibacillary cases occur, and they cannot induce immunity that is sufficient to reduce the number of bacteria. And they have a lot of IgM antibodies. So there is a difference in biomarkers occurring in the spectral site of leprosy, so either falcibacillary or multibacillary disease. However, it is not a black and white phenomenon that multibacillary patients only have antibodies and falcibacillary patients only have inflammatory cytokines. There is, as you can see here, an overlap, and therefore it's very important to test and screen a lot of cases so that we can can get an idea of which biomarkers really are specific for disease and do not occur only in also in infection. And so therefore only not one biomarker will be sufficient to make the difference, to discriminate disease from infection and possibly from multibacillary disease. So we need to identify a lot of biomarkers in a population. So what we have done in the past well, over 15 years is to screen population that are composed of individuals that we know are not infected and do not have contacts with leprosy patients, as well as individuals we know that who are infected or have the disease. So we always use in our research clinical references. So patients and control individuals that we know are clinically patients or clinically not infected at all. So, and we then we screen several biomarkers and look at which biomarkers can make the difference between uninfected and disease, or for instance infected and disease, so that we have biomarkers specific for the stages I just mentioned. And one a very old biomarker was actually the first biomarker for leprosy are antibodies. And you may have become familiar through the past 2 years with the COVID pandemic that after vaccination or after an infection, that people develop antibodies against the pathogen for which the vaccination was meant. And for leprosy, people that have been infected with leprosy can develop antibodies directed against phenolic glycolipid 1, shortened to PGL1. And this glycolipid is the major part of the cell wall of Mycobacterium leprae, and it's composed of 3% of the bacterium. So it's also good to know that Mycobacterium leprae PGL1 is specific for Mycobacterium leprae. It also occurs in a different variant in Mycobacterium tuberculosis, but it's specific for Mycobacterium leprae. So it has been researched a lot and amply described that the levels of IgM antibodies against Mycobacterium leprae PGL1 correlate strongly with bacterial load in an individual, and therefore are also reduced upon efficient treatment of patients. And that is very important to keep in mind, so that the levels of the antibodies correlate with bacterial load. And therefore, this first biomarker of for leprosy, therefore it can indicate Mycobacterium leprae infection and also associated with multibacillary disease. However, it's not so good to detect possibacillary disease because possibacillary patients do not have a lot of bacteria and therefore not have a lot of IgM antibodies against PGL1. Furthermore, what's also good to realize is that the antibodies also do not indicate disease, so they indicate infection, and they can also not be used to predict disease because people that are infected with Mycobacterium leprae do not always develop disease. So these are things that are good to keep straight, so that it can detect multibacillary disease. It indicates Mycobacterium leprae infection, but it cannot discriminate between present and past infection and also not detect possibacillary disease. So we need new biomarkers, of different type of biomarkers for possibacillary disease or early disease stages, and also to discriminate only infection from multibacillary disease. So what we have done the past 15 years is to work on the identification of host biomarkers. And we used several approaches for that, and the approaches are referred to as the Omics approaches. So for instance, genomics, then you identify which DNA, which genes are important or associated with disease. Transcriptomics, then you look at the RNA, and also metabolomics we have used in our biomarker approaches. But here in this discussion, proteomics, so the identification of proteins is important because that is what we have used so far for our diagnostic test development. So what we did is we used several platforms, high-tech platforms that from a tiny sample that was taken, for instance, in the fieldwork in Bangladesh, but also in other countries, this tiny amount of sample could be screened in an omics, a proteomics platform for multiple proteins, for the presence of multiple proteins. I think we have tested over 100 proteins per sample. And we looked at which proteins were different, so the levels were different in multibacillary patients compared to postbacillary patients compared to the healthy controls from the same endemic areas. And we looked at the differences. So the ability of only antibodies to detect multibacillary or postbacillary disease. And what you can see here is that, for instance, the part of the circle, part of the pie that is indicated in green, that is, those individuals can already be detected using antibodies against PD-L1 alone. If you look at the yellow part, which is the majority of the pie for postbacillary patients, then a lot of cellular markers are associated with this disease. So these are new biomarkers that we detected through our research in proteomics. And if you then look at the blue part, these are the individuals per patient group that are detected and using a combination of both antibodies and cellular markers. So what you see here is that the sensitivity is increased. So the combination of using multiple biomarkers for both antibodies and cytokines, that combination of biomarkers improves the leprosy diagnostics, whereas it still remains negative in most of the healthy controls. So the specificity remains similar. So, of course, it's not perfect yet, and you see differences per country. So, therefore, we are still improving that, and I will get back later to what we are doing now with the biomarker platform. So, having identified both antibodies and cytokines as biomarkers for different parts of the disease spectrum in leprosy, that is one thing of the research. But translating this to a low complexity field test is something else. So for that, we have been working together with researchers from another department at the LUMC, and for that we used so-called UCP lateral flow. Well, UCP stands for technique, and technique is upconverting reporter particles, and it's actually like the same as an ELISA, but then a field-friendly method for it. And what is important is that it is a quantitative assay, so it doesn't give you, like for instance, the antigen self-test of COVID or a pregnancy test, which gives you positive or negative. No, this test also gives you a level. So it is positive or highly positive, but there is a level directly associated with that. So there is a number. And this is important. I'll get back to that later. This is important for several use cases of this point of care lateral flow assay platform. Well, of course, what's also very important, seeing the areas in which the tests are supposed to be used, is that they are very stable without a cold chain, so they can be kept at room temperature, and that it's very sensitive. So the upconverting reporter particles are detected in low quantities. So since 2007, I've been working together with Dr. Paul Korschens at the LMC on this platform, and we have published several biomarkers already in their UCP lateral flow assay format. And I will show you in the next slide that how this is done actually. So what we are now able to do is not only take venous blood and stimulate that and use that as a source to determine biomarkers. No, we can already use a tiny finger prick, so only 20 microliters, and take that and put it in a little bit of liquid, a buffer, and add it to a cassette which measures the biomarkers in each sample. And the importance to realize is that, of course, if you have direct blood on a lateral flow strip like this, there is a lot of hemoglobin in it, and there will be hemolysis. But the UCP signal does not interfere with that. Or is not interfered by that. And so we can quantify the results from only 20 microliters of blood, which is very useful for point of care use in the field. And in the next slide, I will show you very briefly how this is done. You see that using a finger prick, 20 microliter, exact 20 microliter is taken by this mini-vet, and that's important to keep the whole assay quantified. That is exactly 20 microliters. It's added to a buffer and lies in that buffer, and then 50 microliter of it is flowed on the cassette, and after the flow, it is possible to read and analyze the signal, so the amount of UCP using the reader that is depicted there. So you do need a reader, but it's a light portable reader that can be connected to a laptop anytime, and that can visualize the peaks that are produced by the sample if a sample is positive. So until 2020, we have been working on one biomarker per strip or per cassette, and allow that to be analyzed by also in fingerprint samples. But what would be, of course, necessary, as I have shown already, that we measure at a point of care level also multi-biomarkers. So because with one biomarker, we cannot identify or discriminate a disease from infection. So we need this multi-biomarker test, and we have been working on that platform for several years and published this in 2021. And I hope you can see it there on the right. You see the strip, which is of course much broader, and this strip accommodates 6 biomarkers and also 6 controls. So per biomarker, 1 control. So this results in 12 peaks that will give you the levels of 6 biomarkers. It is possible to analyze the multi-biomarker strips with the same reader using an adaptation for the cassette holder, and the same way, the same software the multi-biomarker test provides you at a point of care level, now also the amount of biomarkers. And this is important because what I mentioned is that some biomarkers are of interest in detection of post-bacillary disease. Others will be present more in multibacillary disease. And because you remain also at this, with this MPT test, we remain at we remain at a quantitative level. So detection is still quantified. This allows us to do to measure several things and to apply the UCP lateral flow as a multi-biomarker test for several use cases. For instance, a potential application for leprosy would be diagnosis and classification in post-bacillary and multibacillary disease, the early detection of a Mycobacterium leprae infection, monitoring treatment, because the levels of some biomarkers, they will decrease upon efficient treatment, whereas other biomarkers will increase. So a combination of that gives you an idea of how efficient treatment with multi-drug therapy is. And another application would be the monitoring of effect of post-exposure prophylaxis. And post-exposure prophylaxis is treatment of individuals who are at risk of developing leprosy and who are infected with Mycobacterium leprae, for instance, contacts of leprosy patients. And using UCP lateral flow assays, it would be possible to see who is infected and what the post-exposure treatment how effective post-exposure treatment is these individuals. And finally, also another application would be the development, also monitoring development of leprosy reactions during MDT. And these are all potential applications, and we're not at a stage that we can apply UCP lateral flow assays for that, for all of this. So we're working still on that, but there are several use cases that are already ongoing or have been started. And one of them is the elimination, so the monitoring of elimination of transmission. So what WHO currently does is use the new child cases for, of leprosy as a measure to indicate elimination of leprosy. However, a Mycobacterium leprae infection in children so would be more sensitive measure of elimination of infection even. And so if we use Mycobacterium leprae, if we use the antibodies against Mycobacterium leprae, so PGL1 IgM antibodies in children, this would be per definition recent infection, because if we take children of like 5 years old, they cannot be infected for such a long time yet. So it provides like a proxy. So these antibody seroprevalence, so that provides a proxy of the transmission in a certain area. So for instance, if you measure and time 0 and you measure 2 or 5 years afterwards, you could measure the effect in seroprevalence in children of a certain intervention in an area. And this intervention could, for instance, be post-exposure prophylaxis. And then it would affect not only the context, but also the Mycobacterium leprae transmission in the whole area. And this is what you could measure if you measure cross-sectionally in children and young children. So we have done that in collaboration with the Institute of Tropical Medicine in Antwerp and the Institute in Varanasi. And we measured in a door-to-door screening, which was actually a initiative to measure leishmania in children in Bihar in India. And so the LUMC test with a finger prick in these children was performed. And we hope to be able to do similar screening in children with a similar age, so not in the same children with similar age group, and to see whether or not the seroprevalence in that area has remained the same, or whether, so that we can know that whether transmission in the area has remained similar. And in this area now, we saw that 12% of the children was positive. And this is, by the way, similar to what we have seen in other endemic areas. And of those 12%, there were certain children that were high positive, because you can also measure the levels, as you may remember. And from those high, in 2020, high positive children, one of them also developed leprosy in 2021. So this is one use case. So to use the UCP lateral flow for detection of antibodies against IgM in the elimination efforts for leprosy. This, by the way, I refer to as version 1.0. So UCP lateral flow that only detects one biomarker. And in this case, it's Mycobacterium, this antibodies against Mycobacterium leprae. Another use case is a approach, One Health approach, as also version 1.0, so measuring antibodies against PGL1, provided us with a tool to measure infection in red squirrels in the UK that were found to be infected with Mycobacterium leprae and lepromatosis in 2016. And as this is a protected species, we cannot do large studies on that, but we did do so in collaboration with the Roslin Institute in Edinburgh. And we saw that squirrels that developed clinical signs also had an increase in the levels of PGL1 antibodies, whereas those without clinical signs did not have any antibodies. And those that developed clinical signs during our study had did not have any antibodies when we started, but also developed higher levels. So also in animals, it is which may be a source of transmission. Well, I don't think the red squirrels are, but another type of animal could be, and that has been shown in the southern states of the United States, the armadillos. They can carry Mycobacterium leprae, and also for these animals, it has been shown that there is zoonosis by the group of Richard Truman. And we measured in collaboration with the National Hansen Disease Program, measured in experimentally infected armadillos, also the antibodies against PGL1 using our point of care test. And also in those animals, we saw that there is a correlation between the levels of antibodies and their state. So whether they were susceptible to infection or resistant to infection. So these are two other use cases. So, and also this is again a version 1.0 of our test. And a final use case that I want to discuss here is to use the UCP lateral flow to determine who needs extra post-exposure prophylaxis. As you may know, post-exposure prophylaxis with a single dose rifampicin has been endorsed few years ago by WHO, and to be given to contacts of newly diagnosed leprosy cases. And however, we don't know what happens directly with the immune response of the individual when they receive a single dose of rifampicin, and moreover, whether this is enough to eradicate the leprosy infection. So if we use an extra test to determine whether the infection has been cleared by a single dose rifampicin, or whether this is not the case and the person needs extra post-exposure prophylaxis, for instance, a PEP++ approach, then we can use the UCP lateral flow to determine to determine directly the effect on the immune system, on the immune biomarkers after a single dose of rifampicin. Well, this is a study that is currently ongoing in collaboration with the TMI Bangladesh, and Nofamari. And so the aim is that we monitor the effect of either SDR, SDDR, so double dose of rifampicin, or an extended regimen, the PEP++ regimen, in newly diagnosed, in context of newly diagnosed multibacillary patients. This is what I refer to as version 2.0. So this means that we haven't exactly determined the usefulness, of all the biomarkers that we're testing. So we test 6 biomarkers, and we need to know through this study which of the biomarkers is useful for this type of use case. Yeah, so every use case may have a different combination of biomarkers. So we test them as single markers, but of course the eventual idea is to select those biomarkers that provide the best information on the use cases we are investigating, and to develop that then as a multi-biomarker test. So together, so that the optimal biomarkers can be detected together on one single lateral flow strip. So to wrap up a little bit, the summary of our research on immunodiagnostic tools for leprosy over the past almost 20 years. So we, of course, always start with where the need is. So that is with the patient, and that is also the active case finding that is done by our field partners. So field sampling and diagnosis, clinical diagnosis and biobanking are key there, and collaboration together with the field is very important. And the next step then is that we train the field and laboratory staff with using our knowledge on immunology and on immunodiagnostic tests, so that they can use it for further research. And the samples that we are obtaining through the field sites have been analyzed through multi-omics approaches, which is still part of our current research also, to identify new biomarkers, more specific, more sensitive, more apt to be applied on the lateral flow test. So this is a current continuous effort that is done with field samples. And after those omics, the best candidates are selected, and depending on the use case, put into a further diagnostic test development that is field and user friendly, and of course also can be applied globally. So that a biomarker profile in Bangladesh can also be useful in the same type of diagnosis, same type of use cases in Brazil. And finally, we make the cassettes, as I showed you in this short video, make the cassette in an FDA approved company, so that every sample can be tested on one cassette that is similar to all others. So for reproducibility, that's important. This slide does not include implementation yet, and this is an overall overall summary of our research. But you may wonder where we are with our research. Well, as I said, there is a continuous biomarker pipeline from biomarker identification in the field for various use cases, either for reactions, PB or early disease, MB disease, or for infection. That is currently still ongoing at several sites worldwide. And this biomarker pipeline, so to speak, feeds continuously into the test format optimization, because we need to optimize or adapt every biomarker to the lateral flow format, because every biomarker is unique and may need different conditions. So the biomarker identification, global evaluation, and finally implementation are all steps that are also part of the research. And one version of a biomarker may be, at this stage, so biomarker identification still, whereas version 1 is already a global evaluation state and only needs the implementation. So that is the acceptance of healthcare institutes throughout the world to accept and implement in their regular healthcare for leprosy. And finally, of course, what is also important in test development and test optimization is what I already showed you, so that we can go from one biomarker to multi-biomarkers, but also the cassette that is now plastic and is contained in a plastic pouch. We would very much like to make that more sustainable and replace the plastic cassette by something else, or the plastic a bag in which is kept by something that is more sustainable and more environment friendly. And finally, what we also want to do is adapt the reader and the software in the reader, so that in the field it is even more user friendly and gives you directly a result whether or not the person has leprosy, because now we work, of course, with biomarker levels, which require then by the researcher in the field a certain knowledge on whether this person can be interpreted as a leprosy case or not. Well, we want to change that software at a time point in which we can do that, in which we have validated globally the multi-biomarker test sufficiently, so that we can say, okay, yes or no in the field. And so these are all steps that are part of our biomarker, of our leprosy biomarker-based test development. And it is a long way, has been a long way, and of course we would not have been able to do that without substantial funding and continuous funding of several organizations, including, of course, the Leprosy Mission International, but also LRI, NLR, the European Union, and Malta Lab. And more importantly even, are the researchers with whom we have done this. And so I want to thank, besides my own team, also the people at Erasmus University, and that were led by Jan Hendrik Vichardes, and the TLMI Bangladesh team, and Diana Hege in Nepal, Marcelo Tavramira in Brazil, Claudio Salgado in Brazil, John Spencer at CSU, the team led by Linda Adams at NHDP, and of course for the Red Squirrel work, the University of Edinburgh. I thank you very much for your attention, and I'll be very happy to answer any questions.
Thank you very much, Annemieke. That was a very interesting presentation and very, very informative. We do indeed have a number of questions, which you might be able to see in the panel, but I will take a couple of them for you at the moment, then you can answer them. Would the presence of other diseases influence the result of this type of test? That's from Ariana in London.
Sorry, I don't see it in chats, but
it's in the Q&A section.
Oh, it's in the Q&A. Okay,
yeah,
okay.
Yeah. So would the presence?
Yes, definitely. That's a very good question. And as you may recall, I discussed the several stages of disease in my presentation. So the stages of disease are, for instance, influenced by helminth or comorbidity. And but what you do have to keep in mind is, of course, the use case of the test. So when are you going to use the test if it's available in your institute? So mostly people who are suspects of leprosy, they have also certain symptoms. Yeah. So what we are currently also including in our study in Bangladesh is other dermatological diseases. So other diseases that may be causing similar symptoms, so that we can then, for instance, certain infections with fungal infections, so that we can take into account that the biomarkers may overlap, but for instance, that the levels of certain biomarkers are higher or lower. And therefore, it's so very important that we do a lot of screening, but also a lot of screening of control groups, and especially control groups with other dermatological diseases. Yeah. Of course, what we also do is we work on TB diagnostics, and we also use these biomarkers for TB research. But then again, the person with symptoms of disease will not be the same, and as so that person can already clinically be very different.
Thanks, Annemieke. There's a related one from Khorshid in Bangladesh. Do you check or have any information that any opportunistic or saprophytes microbacterium can influence or trigger the biomarker level?
Yeah, I think the same answer applies here. And as we discussed last week, that week before with the TLM team, the inclusion of other diseases that may initially look like leprosy is very important to screen for biomarker levels. Yeah.
And in the chat, Dr. Ari from Mozambique has asked, would immunofluorescence add any value to making tests more sensitive or creating other observation options, for instance, in combinations with other technologies like spectral imaging?
Yeah, I think that definitely it would be important to combine. I always refer to our test as adjunct diagnostic tools. Yeah, so by itself, they don't provide the complete picture. You need everything that is useful, and therefore the question you want to answer with the test, so the stage at which you had, so it could be like a triage test, and then you would, say either no leprosy or potentially leprosy. So you would refer that individual maybe for something with another extra test, so that you get an even better diagnosis. Does that answer the question?
I think so. Ari can come back to us if not.
Okay.
Me. We have a couple of questions from the Leprosy Mission's fundraising offices. So one is from our Netherlands office from Henno Kupri. How can the Leprosy Mission support this research? And maybe if I can combine that with the question from Sean, and there's a related question from Elias as well. What's the amount of funding that we need to take this forward and to actually speed up the process? What does it translate to in terms of an annual budget needed?
Yeah, I always find it difficult. I would first start to say what is needed. If you keep in mind the slide in which I showed the several steps involved in biomarker or in test development, what would be a next step is that to perform similar research in so what we're doing now in Bangladesh in other countries. And then what I mean then is exactly copy paste. Yeah. So what is very important, that there is a good team in the field for diagnosis and for clinical diagnosis, so that is the reference group that you wanna have. So the amount of efforts going into active case finding, treatments potentially post-exposure prophylaxis treatment of contacts, and taking samples and doing the same amount of evaluation, because you have to know that in the since 2012 we've been working together with a team in Bangladesh, so that is 10 years already at different stages of our development. So and the amount of samples that they have recruited and taken is enormous. Yeah, but this is needed because there are so many variables. So what we just discussed, well, potentially co-infection, or if you only have leprosy or leprosy infection, there is already the complete spectrum. So what would be needed is to do same kind of recruitment, sampling and testing, and comparing the same biomarkers to see whether they hold true of what they're capable of doing or discriminating in Bangladesh, and same in other countries. That would be a logical next step. And in Bangladesh, where a lot of this has already been done at this moment, we are validating the biomarkers in the use case for treatment of single-dose rifampin. So that has to be completed along with a lot of testing of well, differential diagnoses. So non-leprosy cases that look like leprosy, to see which biomarkers and the levels of which biomarkers are useful. Because this brings me back to a question I receive very frequently. So what is the sensitivity specificity of an assay? Well, it's not like a qPCR, so that you know, okay, there is bacteria, and how well can you detect it, or how many patients can you detect it, because there is no gold standard for a leprosy infection. And that is because we have this spectrum. We have these post-bacillary cases that do not have the bacteria, they do not have antibodies. So we want to identify in that early disease or in post-bacillary disease, both of those stages, we want to identify biomarkers without having a gold standard. So you may envisage it needs a lot of samples and a lot of validation. And so that is the logical next thing. And I always say also for other studies, start by biobanking, start, sampling. That is something, if we don't get the funding to do directly all the same things that we're doing in Bangladesh, at least you have all the sampling and you have the clinical diagnosis that would already be part of your healthcare and would already be part of supporting the countries. Then you would already do that. And then at a certain stage, evaluation or validation can be performed in other countries.
Thanks. I can realize we're fast coming up to the hour, but we've talked about at the moment we're still in the studies and we're taking lots of samples. So the actual cost at the moment may not be the final cost. But there's a follow-up question from Eliash. Is there a target cost or price per test, and where are we in that process? I mean, you showed the video of how easy it is to do. But actually, what will it cost? Because for it to be field friendly, it has to be affordable to national programs. So what are we aiming at?
Yeah, well, if there's only one biomarker, like we have now, it is between 2 and 3 euros. And you may want to know that packaging is a huge part of the cost. So that is also something else that the sustainability, the environment, that is a very important part. That I am looking for other opportunities to lower the price while keep, the same. So, but keeping the cassette in mind, so the plastic cassette, plastic holder with a strip with one biomarker, and that would be between 2 and 3 euros. Yeah.
Thank you. Thank you. We do have a few other questions that are in the chat, but I think for the sake of time, possibly we'll answer those in a different way. I can see they're coming mainly from it within TLM, so we can find a way to answer those. Annemieke, I just want to thank you very, very much for your excellent and very clear presentation. And my understanding has increased greatly, even though I've been watching what's been happening in Bangladesh for many years. Yeah. And I'm sure other people's understanding has increased a lot. Thank you, everybody, for joining us for today's webinar. I hope this will be the first of many opportunities to learn what Annemieke and the team in Leiden are doing, and the other partners that are working on this very important subject. We will share the recording very soon, and any questions we haven't managed to answer live, we promise that we'll get the answers to you. If you want to know more about this subject, you can contact Annemieke directly. Her email address was at the bottom of all her slides that she showed, and it's a.geluk@lumc.nl. If you want to know more about the research that the Leprosy Mission is involved in, you can contact us at research@leprosymission.org. And with that, I'd like to say another thank you to Annemieke and another thank you to you all for joining us. Thank you very much.
Thank you.